Okay, today I'm going to talk about antigen processing and presentation. So first of all, uh, any pathogen, whether that is bacteria, fungus, or virus, that has to be processed before it can be presented to cells of immune system, and cells of immune system, particularly the T cells. Okay, T cells. And we have two kinds of T cell, if you remember, CD4 and CD8 positive T cells primarily, which are taking part in immune response. Okay, so uh, for antigen presentation we have two pathways. Antigen presentation, how many pathways? Two pathways, and those pathways are called as exogenous pathway and another one is called as endogenous pathway. So we'll talk about these pathways today in this lecture briefly. Okay, so let's here. So we talk about exogenous pathway first. Exogenous pathway, for suppose bacteria. So this is a cell and you have bacteria outside here. Now bacteria can gain entry into the cells. For example, exogenous pathway, you have an antigen presenting cell here. Antigen presenting cell, let's consider it as a macrophage. So bacteria, this cell would take up the bacteria through phagocytosis. So once the bacteria is inside the phagosome, so these are bacteria inside the phagosome, and then you have lysosome here. Lysosome is rich in acidic proteases, and its pH is acidic, so it can fuse. So phagosome and lysosome would, and they'll make it phagolysosome. So fusion, these bacterial peptides this bacteria would be broken down into smaller peptides in the phagolysosome. So let's now move on to the another aspect here. In the cell you have endoplasmic reticulum. So this is rough endoplasmic reticulum. As you know, rough endoplasmic reticulum is rich in ribosomes and there is protein synthesis, and in the endoplasmic reticulum there is assembly of different proteins to make a functional molecule of protein. So here in rough endoplasmic reticulum there is formation of MSC2 molecule. So MSC2 molecule, if you remember, it is a molecule looking like this, consisting of alpha and beta chains. So this gets assembled inside the MSC2 molecule, and then through Golgi apparatus it is transported across the Golgi apparatus finally to in an endosome, finally to fuse with the phagolysosome. So how it is happening? So this MSC2 molecule is also attached with a invariant protein, II protein. Okay, so it moves here through the Golgis, and this invariant protein is also responsible for the movement of this. This acts as like a chaperone. So once this molecule is inside this Golgi endosome, then this clip molecule, the II molecule, this chain here is degraded. Let me put it little bigger. So this only the part of the head part of this invariant molecule will remain, and this the tail part will be degraded, so it is still binding to the groove. So the purpose of this binding to the groove is that no other protein or peptide can bind to this cleft, because this cleft is meant to bind with the peptides, which are the bacterial peptides in the phagolysosome. So it goes inside. Once it fuses, and then the peptide can be loaded onto it, and the clip peptide is removed, and the bacterial peptide or the antigenic peptide is loaded. And there are other molecules here. For example, HLA molecule is responsible for this process also. And finally, the peptide will be displayed on this MSC molecule on the surface, and then the T cell, the one with CD4 receptor, can recognize along with the TCR. So there will be then this CD4 will become activated, and they'll become like T helper cell, and then they start secreting different cytokines, which will be responsible for developing the immune response, maybe activation of B cells or so on. Okay, so this pathway is for the exogenous antigens. This is for exogenous antigens like bacteria or fungus, which enters from outside into the cell. So when they are entering from outside into the cell, they are processed through this pathway here: phagosome, then phagolysosome, and then MSC2 comes and binds with the peptide, bacterial peptide or the fungal peptide, and then it is displayed on the surface of the antigen presenting cell. In this case it is macrophages, and then the CD4 binds with the along with the CD4 along with the T cell receptor TCR. TCR recognizes the antigenic peptide, CD4 recognizes the MSC2 molecule. So these antigen presenting cells or the CD4 cells are MHC2 restricted. That means they can only recognize the antigen when it is presented along with MSC2 molecules. So let's talk about endogenous presentation, endogenous pathway. Endogenous. This pathway is used by the cells for processing and presentation of antigens such as virus, maybe cancers, antigens, intracellular bacteria such as mycobacterium, and then even the autogenous antigens, the intracellular ones. So think of a cell here. You have some virus entering the cell. That virus, uncoating happens of the virus, and then obviously the replication of virus would take place by utilizing the host machinery. Some proteins would be synthesized, and some of these proteins, they get ubiquitinated. Ubiquitination is a process that leads to that prepares the proteins for degradation through proteasome. So ubiquitination in proteasome is a mechanism to degrade the proteins, non-functional proteins or some other error proteins, and then these are broken down into small peptides, around 8 to 10 amino acid long peptides. These peptides then can enter the rough endoplasmic reticulum through transporter associated antigen presenting protein TAP. It is a transporter, so these peptides can get inside the rough endoplasmic reticulum and then they can bind with MSC1 molecule. MSC1 molecule, the groove, antigen binding groove of this alpha chain here, alpha 1, alpha 2, alpha 3, and this is beta 2 microglobulins. So these peptide can be loaded onto this antigen binding groove here, and there are some chaperones here that helps in doing so. These chaperones are calnaxin is there, calreticulin is there, ERP57. Beside this TAP there is tapacin that helps to move the MSC molecule close to TAP, hence allowing the peptides to bind onto the groove. And these chaperones here, the calreticulin and ERP57, helps to properly fold the MSC molecule, and then this MSC molecule, MSC1 molecule associated with the antigenic peptides move to outside the surface of the cell with the antigen binding to the groove of the MSC1, and then it can be recognized by the CD8 positive T cell. So CD8 positive T cell will have TCR receptor here that would recognize the peptide sitting inside the MSC molecule, and CD8 would recognize the CD8 receptor would recognize this MSC1, whether this is MSC1 or not. If it's not MSC1, obviously the CD8 won't recognize it, and there will be no TCR won't be then binding to the peptide here, antigenic peptide. So once this recognition happens, the CD8 cells become activated, and then they become CTL, cytotoxic T lymphocytes, and then they can release their granzymes and another toxic granules into the affected cell or the altered cell. This is called as altered cell because it is infected cell, and then this is destroyed. Finally this cell will be destroyed. So this is how the intracellular pathogens, the bacteria, or the viruses, they are, when they infect the cell, then this is how the immune response would start. After this, all the infected cells would be destroyed like this, and obviously beside this there will be the activation of T helper cells could also take place. Certain cytokines would also be released by these cells, but very few cytokines are released by CD8 positive cells. But ultimately this will result in the destruction of the infected, viral infected cells or the cancerous cells. That is how the immune response takes place.